was slightly disappointed in grading the test. So I don't know how. I haven't done the statistics. Normally in the US you have to give the statistics of the test, but out of 30 points, I think the maximum scores was 25, and most of the people were between the 15 and 20 range. So percentage wise, it's like 50 to 60%. So that is why I was not very happy. But anyway, the whole purpose is not the exam. The purpose is getting you interested in the topic. So, again, at the end of the class, I think Professor Das would give you back or at least show you your answer sheets. So, no course in nanotechnology or for a new topic, for the new area is not complete unless you know whether it's perfectly safe or what impacts it has on the society. Nanotechnology is a new area. It is slightly controversial because the reason it got more controversy is I forget the name of the author. The person who wrote the book Jurassic Park. Have you? The Jurassic Park is a movie. The person who wrote the book of Jurassic Park, the same person has written a book on nanotechnology. I don't know whether anybody have you read the book? If you read the book, he actually looked at the self-assembly section of nanotechnology. What is self-assembly? Is the nanomaterials keep growing, assembling and growing it. So he wrote that the self-assembly goes out of control. So that was his story. So he called it the nanobots. Nano like robot, he called it nanobots. So nano BOTS, and basically the whole story is about how much havoc the nanobots create to the society. Like they grow out of control, they become like Frankenstein. You know, have you read Frankenstein? The Frankenstein is created by a scientist, but then Frankenstein goes out of control. So that book became a bestseller in the US. It hasn't been converted to a movie yet. I don't know where, someday it might be. So that created people's lot of concerns on nanotechnology. Before that, people, general people had no idea what nanotechnology is, but now it became a very controversial issue. The same thing as genetic modified foods face in Europe. The same reason is people still have a feeling that the genetically modified is not good for human consumption. Again, I don't think there have been conclusive evidence to show that the genetically modified thing is not good for human beings, but that is people's perception. So that's why we have to talk about what impacts nanotechnology would have, because it's kind of what we call, I'm trying to look for the word in business. These are called as not incremental technology. There are two types of technology. One is the incremental technology, that the other technology is up to here, it makes slightly better, and the other technology is just takes things off. For example, digital photography is a technology which basically shuts down the other technology. Using of computer CAD totally took away the draftsman and all those things. So nanotechnology, there's a term in business for that. I'm trying to remember. Could you repeat it?
They call it disruptive technology.
Disruptive technology, that's right. So nanotechnology is a disruptive technology. So if it goes the same thing like artificial intelligence, that's a disruptive technology. Suddenly if artificial intelligence comes in, lot of people who are doing regular stuff would be replaced by technology. For instance, people who drive cars, drive vehicles, they might be obsolete because it would be. So that is why the societal impacts come in. Now, also the other issue is, as you know, the nanomaterials are extremely reactive, and since they are extremely reactive and also very, very small, our human body works with a filtration system. So, with that filtration system, I have the presentation over here. I am actually telling you because today we do not have a lot of time. So, I am trying to tell you the overview. The presentation is with you. Today I will not go through the presentation like I have done in the earlier days, because I do not think we have time enough to go through every slide. But in a nutshell, if the nanoparticles are extremely reactive and they are very small, how small they are? You know, in body there are filtration systems. For example, the air you breathe in, it's filtered through some nanofibers. Those nanofibers are present inside our nose, and they adapt. People living in polluted areas have more cilia than people living in non-polluted areas. Okay. So, because the body kind of tries to adapt and try to get, but that is not enough to stop nanoparticles. People, when they talk about pollution, pollution is not nanoparticles. Nobody knows how much nanoparticles are there in pollution. You can't measure it, and so nanoparticle can get into the human body. It could be getting, we are talking about 3 different ways. One is if you touch it, second is if you breathe it, and third is if you some way ingest it, that means you eat it. The touching nanoparticles, people wear gloves to touch nanoparticles, but even if you did not wear gloves, we talked about surface tension, capillary action. So, the pores in the skin, even though nanoparticles might be in the order, I think we showed a picture of where we showed the hand, the very first slide where we showed the hand, and progressively we magnified the hand as we went into the nanoparticle size. Nanoparticle is lot smaller than the pores, but again, because of surface tension, and also our body has pressure, blood pressure, and blood pressure pushes things outside. So, the chances of those particles leaking in and getting into our bloodstream just by contact is minimal, unless they have been put in by intention. But still, does it mean that we should not be wearing gloves? Yes, we should be. Why? Because if the nanoparticles are there, you wash it, do whatever with it, it still stays. Okay? You can't get rid of the nanoparticles. So, next time it might get into your digestive system, because next time when you eat, it might be a part. So, that is the reason you should be wearing gloves, but gloves is primarily preventing particles from getting onto the. If you ingest it, there have been results where people have shown digest it. For the human body at least, nanoparticles, they are just get out through the other end of the body. They do not, unless they get into the bloodstream, and normally they do not, they get out. But if again the result is not conclusive, one of the reasons the result is not conclusive is there is no way we can monitor. We have put 100 nanoparticles from this end. If we count, if we get 100 particles coming out from the other end, unless we do this, we cannot be sure, because it is difficult to count. We look at the density, how much we there, we look at the density. So, it could be we lose some nanoparticles. So, once if it happens, it might be okay, but if it happens on a regular. So, that is the part of work which I was doing. I was working with the Connecticut Department of Agriculture and US Department of Agriculture. One of the applications of nanoparticles, well, I will be going to there. I will just talk about the third place how the nanoparticles can get into the body. That is through if you breathe in the nanoparticles. That is the worst possibility, because nanoparticles get into all of the filtration mechanisms, and it finally lands up inside the lungs. And so, people have seen that. So, depending on the size, unless here in the toxicity case you want the nanoparticles to agglomerate. If they agglomerate, they form bigger chunks and your body can throw them out. but if they still remain in their nano form while they have come in, then they accumulate in the lungs, and when they stay in the lungs, obviously through a period of time your lung capacity decreases. It's like smoking, the same effect. If you smoke, then basically you are putting in again, when smoking also, you are intentionally putting in nanoparticles inside your lungs, because the carbon when the combustion of the end of the cigarette happens, it produces very small bits of particles, which are the most dangerous part of a cigarette. That is why again, it is, I am not saying that, but people have said that there is a concept known as e-cigarette. In e-cigarette, what they do is they make the combustion. Normally in a cigarette smoke or any smoke, combustion is you have 40% efficiency. You can do thermodynamics and you find that 60% of the energy is wasted and they produce particles. So, if you can make the energy more efficient, so in the e-cigarette they have a monitor, they have a temperature sensor which regulates the kind of temperature with it. So, there is a complete combustion. So, there the nanoparticle intake has been reduced, but nicotine is still there, that is a part of it. So, whatever harm that causes, that is still there, but at least the smoke part has been reduced. So, anyway, so nanoparticles get into the lungs, and if it gets into the lungs, we do not know, there haven't been anybody who has been diagnosed with something like cancer or something. Even if they have been diagnosed, there is no conclusive proof or evidence saying that the person is caused because of nanoparticles, because cancer can be caused by all different sources. Again, it is very difficult to do a control study. You have to do animal study, you have to do control study to kind of know, but it is so difficult to measure controlling. So, that is why the toxicology results are. So, my work on toxicology hasn't been with animals. My work in toxicology has been with plants, because that is one of the main sources of where we can get in nanoparticles. Why? Because where the nanoparticles are used, one of the most important components of fertilizers is they put in the nanoparticles. They make the fertilizer better. They make, in fact, even certain hormones are being put in nanoparticles and put into the plants. Yesterday, I was talking with one of my friends, and they were telling that he owns a farm, and there, to kind of make the size of the fruits and vegetables better, they inject extra hormones in over there, which very quickly make the things grow. So, how to make these hormones more efficient is by administering it with nanoparticles. So, there have been two areas where people are work in the area of toxicology. One is the area of engineered nanoparticles. Engineered nanoparticles means the particles which artificially you have created, you have put into the materials, and then there are natural nanoparticles. Natural nanoparticles does not mean the source has to be natural, but those are things that come out from the exhaust of your car or come out from the chimneys of the factories. Means those you haven't intentionally done something to change its properties. So, engineered nanoparticles are the worst, are most I would say for the living beings. So, I will be showing. So, this is that's the reason that comes in over here, the ethics. So, you want to make your fertilizer more efficient, definitely. You would like to put a little bit of fertilizer, you would like to make your fertilizer grow so well that your plant would grow bigger, you can make your agricultural production better, but not at the expense of human health. So, bigger tomato, if it is not a safe tomato, that is the same concept with genetically modified. So, again there has been no results to show that the engineered nanoparticles are responsible, because normally lot of these effects are long term. So, studies have been done with animals. I did not take part, I did studies with plants, and I would show you some results of that, of how much the toxicity. So, that is what the ethics come in. So, this is something you have to always keep in mind. The quest of science should not be the only goal. Also, science is betterment of human civilization, but you have to also look at the negative aspects of it. We have seen that when technology comes in, there are positive effects and there are negative effects. Alfred Nobel, the guy who made the Nobel Prize, do you know what he discovered? The dynamite. So, he used it for making mining easier, but then that has consequences. Internet when it was initially discovered, it was discovered by physicists in the CERN lab, and they wanted to communicate, exchange data across continents. It was excellent, but now along with it has come a lot of other effects. So, that is why it comes of the words ethics is the scientist has to be warned of the ethics, means so that warned of the harmful effects, so that they can use it properly ethically. So, that is the talking of ethics of nanotechnology. Again, I wanted to be these are some of the results which we show is again studies have been done with fishes. Again, one of the reasons is in the US there has been people, they have, because of toxicity, again this toxicity that here it basically shows the gill filaments of a zebrafish when it is exposed to copper. Again, one thing you should remember, all these toxicology studies have been done in a very amplified. So, it is very rare that a zebrafish would be exposed to 50 nanometer of copper nanoparticles for 48 hours, unless it goes into a place like a copper mine or something. So, that is what you have to be careful of. One thing that is positive about nanoparticles or nanomaterials is we only use grams of it. So, by the time we look at these type of concentration or these type of effect, continuous prolonged effect, it has to be, but not for the general public. You have to look for yourself, because a scientist who is working in this area is exposed to nanoparticles lot more than everybody else. So, that is something you have to take care of yourself. You are, whenever you are working with silver nanoparticles, you are inhaling some of them. When you are working with any other particles, you are inhaling. I think you also know that the person who discovered radiation, Madame Curie, she died of cancer. She got exposed to radiation because that time we did not know that it was so much harmful to it. So, we do not want the same thing to happen to people who are dealing with. So, that is also another reason you have to be. Again, how you would be careful, that we still do not know yet, because any standard, people will say they wear masks to work with the nanomaterials, but if you look at nanomaterials, masks are actually useless. They would just penetrate through it. I actually talked with I think Rohit, he had a design of making a better mask to prevent, if some better things come out over there, then maybe it will be, but there are better agents which you see when people go into the hazmat areas, when the people go up in the toxic areas, they wear these special type of masks. Those have special filters which work with. You would need to have electrostatic filters to stop it. But whatever I am saying is the fish consumption in the US has decreased, especially for large fish, because people are scared that what happens is the fertilizer nanoparticles, what it gets washed by rainwater, and ultimately it lands up all in the ocean. And most of the fish is not caught well deep into the ocean, but it is caught kind of in the shallow ocean water. And what does the fish consume? Whatever the material is available, and it has been observed that the fish has a high concentration of nanoparticles, and they get concentrated inside the fish. It might be not as dangerous for the fish, but it is there, and so if that fish is consumed by human beings, it gets transferred. This is so this is what they say: zebrafish will ingest small particles, and it shows wherever the. So these experiments are shown that where you can see these nanoparticles showing over there. Particle concentration with time, how much the nanoparticles kind of when they disperse, how much coagulation they do. This is also important. Aerosolized nanoparticles. So aerosolized nanoparticles means basically nanoparticles which are sprayed out. So these are the experiment. If you release it, people are seeing how much effect. Again, there has been no direct experiment, but I said is how much effect it has on the lungs. So when the particles come in, when it goes, they were showing what is the particle diameter, and based on the particle diameter, this is the total number of particles, how much it gets deposited. Let me go. You can look through these. I'll go down to my own research, which is a little later down. Catalyst of the carbon nanotube, which has the main problem. Let me go down. So again, I mentioned it a number of times, is nanotubes have been thought to be toxic. The main reason they have been found to be toxic are because of these metal particles present in over there. So if we, because those are the residues, the residue material which stays and which creates, if it's done on a support, a lot of nanoparticles are grown on a support, fine alumina. Asbestos was considered to be a safe material. Still right now it's one of the materials will people avoid. Again, that is because of this fine alumina which is present, fibers which are present in asbestos, and asbestos land up into the in the lungs, and so it creates all sorts of breathing issues. For fibers, it stops better. Nanoparticles are even smaller than that, so the effect is not as quicker or as much as asbestos does, is because you need to have a little more of the nanoparticles get in before it starts getting exposed for a longer time. So there we are we have in a better condition, but this is they talk about with the fiber exposure, what are the different types. You have short fiber. If you have long fibers, what happens to the lungs? Let me move over, and actually I will try to. Quantum dots. This is cadmium selenide. So if it's bioactive, you are making it bioactive to get hold of proteins. So the same material, when it gets into your stream, it's still bioactive. So it would react with some of the proteins that present inside animal body, and that might again lead to issues. So this is one of the big problems which the environmental agencies are trying to do, is what happens in the food chain. As I said, the nanomaterials used, or even in the lab, after you have used, you put it in the garbage is taken over, it's dumped someplace else. From there, it is again, you might think it's a very small amount, but the thing is, because it reacts more, So, it eventually lands up into the food chain, and so that is why especially it lands up more in the vegetables than, in fact, from the point of view of, and that is one of the in the US, and right now I think even in the India there is a fad of having organic food. So, organic food means food that it is grown without the help of any pesticides, any what we call fertilizers, any chemical fertilizers. Okay. But what are they grown on? They normally they are grown on soil which is very fertile, and where does this fertile soil comes out from? Most of the time it comes out from the garbage, means wherever the garbage is recycled, it is over there. That is where the fermentation happens, because the soil has to be rich in nitrogen, and that is where you have the nitrogen rich soil, and those are the soil on which the organic food is grown. But those are the places where through leaching and all these effects you have a high concentration of nanoparticles. So, organic food has higher concentration of nanoparticles in them than food which is grown in artificial means, because you are intentionally growing on the soil which has more nanoparticles in them. Again, nobody knows whether that is good or bad, but that is a point of contention against organic food. This is, let us where it comes in the inside the lungs, where it kind of gets. This is the ear, skin. So, ethical lessons: people do not want nuclear energy, people do not want the genetically modified food, other. So, asbestos. So, that is one of the issues which is taking place of nanomaterials. So, this is public perception of the technology. It came out with from, I think from that book. Do I have the mention of the book? There are people who are working with making it more popular. Okay. This is actually a presentation about our work given by my collaborator in this work. He is the chief scientist and the director of this. It's known as the Connecticut Agriculture Experimentation Station. So, they were responsible in finding the West Nile virus. The other ones means lot of these things that affect agriculture. So, so I will just go briefly over the presentations. Today we have to end at 11, isn't it? So, would there be a break, or it will go continuous, I presume, because anyway we started late, isn't it? So, no break. So, we will go and whenever we. Okay. So, these I said artificial nanoparticles. So, these are. So, this is the EPA definition. Nanoparticles have at least 2 dimensions less than 100 nanometers. Nanomaterials, this is again, it is little bit different from what we have been taught, but in the Environmental Protection Agency, what is written in their thing, that is what he says is nanomaterials have at least 1 dimension less than the 100 nanometer. This actually initially started with 250 nanometer, now they are lowered it down to 100 nanometer. There were expectations they might be lowered down more, but right now with the new government, the EPA may not be existing. So, that is why we do not know what is going to happen. So, where the nanoparticles are generated, it is generated wherever there is a fire, a forest fire or something fire. If there is an explosion, it goes in volcanoes, other again the human activities. These are what we produce, natural nanoparticles, not engineered nanoparticles. Coal combustion, vehicle exhaust, the weathering of rubber tires is in fact one of the highest concentration of metallic nanoparticles are next to highways. So, people who live next to roads have a higher instance of ingesting inhaling nanoparticles, because for the vehicular exhaust, that is especially there have been a study in Connecticut made by, they took the soil from the next to the highway versus the soil 50 meters away from the highway, and they found it was a huge difference of the nanoparticle concentration. One of the things that's used in car catalytic converter is cadmium nanoparticles. They are used to make your combustion better. I don't know exactly what it does, but it's there in the catalytic converter of cars. Currently, every car has a catalytic converter that increases the car's gas mileage. It makes the combustion more efficient. And there is a company in Connecticut who basically digs up a little bit of the soil from the area just next to the highway, and they filter it, and they make their cadmium nanoparticles from there. So there are a lot of there are similarly there are companies which buy old motherboards and computer parts. Why? Do you know why you get old motherboards and computer parts? Gold. They extract the gold from over there. So these are the engineered nanoparticles. The engineered nanoparticles are we have been trying to engineer, means doing something to that, and this presentation was old. So nanotechnology was a 1 billion industry in 2005, and it says it will be a 1 trillion industry in 2015, but I think it is more than 1 trillion currently. One of the reasons is a lot of the industries and nanoparticles are not accounted for, because industries don't have to make public, and they intentionally don't make public if they have added nanoparticles to it. As I think, as I said, in the salt and sugar, nanoparticles are added. Equal people who use sugar substitute, that has a tremendously high concentration of nanoparticles compared to regular sugar. So there are reports which people say that if you are diabetic, you should not have sugar, or if you have to have sugar, have regular sugar, don't have equal or the sugar substitute, because the sugar substitute is basically they want to make the thing by applying more, and so more nanoparticles have been added. This is the just definition of how nanoparticles change. So what are the nanoparticle based products? There are different products. We talked about the graphs which shows different sunscreen. Nanoparticles are present in lipstick, it's present in fact one of the largest consumers of nanoparticles is Lakme. So they use nanoparticles for a lot of, historically Indians have been using nanoparticles for a long time. That is the mascara which you put it on your eyes, that has carbon. In fact, if you look at it, it will have a percentage of carbon nanotubes also. It has a lot of the carbon nanoparticles, and historically the reason why people put mascara was thought to be it was good for your eyes. I don't know whether there has been a study to show that. So again, one thing you should be knowing is nanoparticles are always considered to be bad. There are, and our data would also show that there are certain instances when they help. So he's talking about nanoparticles in agriculture. So nanoparticles are components in pesticides and fertilizers. For the most part, there are no labeling requirements. No fertilizer advertises saying that they have nanoparticles in them. So nanomaterials often fall under the they have to put a label. They have to say what are the materials which are present in over there, and they have another category known as inert ingredients and or other ingredients, and they put everything over there, and the nanoparticles have been put in that category. So EPA had started a policy in 2011 to seek information of nanoparticles in pesticides. the focus has been primarily, there have been few nanoparticles which have been focused. Silver nanoparticles is one of the ones which the EPA is after, because silver can be added as to promote shelf life of materials. So, right now people want to, there is lot of nanoparticles which are present in packaging. So, when you store materials in shelves, you would like to have the material stay there for a longer period of time. So, one of the things that they are put, they do not want bacteria to get in over there and damage. So, whenever you put a polythene wrap on top of the materials, that polythene wrap or whatever has silver nanoparticles put in over there. So, if you are getting things to increase shelf life, so that is a place where a lot of nanoparticles make in their way. In fact, he had a project where he was working with burgers from Burger King, and they had a lot of nanoparticles, because everything McDonald's, Burger King, when you buy, they kind of wrap it and package it with the material. Intentionally they have put in silver nanoparticles over there, so that the material won't have any bacteria in it. Again, nanomaterials, there is a question, are nanomaterials an emerging class of contaminants? It is not contaminating, but that is so yeah, yeah, so did I tell you about the nano water particles? Recently I didn't, it's not, I haven't put it over there, is there have been, there's a technology where nanoparticles have been used to, when you get fruits, vegetables out from the market, how do you try to get rid of if it has bacteria on top of it? If it has pesticides, it's a different question, but if it has bacteria, how do you try to? You wash it in water. But again, washing in water, we talked about biofilms, other materials which stick very much onto that. Washing in water does not do anything to them, they are still over there. You have to wash with pressurized water, maybe, still that nobody washes the materials in pressurized water. Even if you do that, it does not. So, these people at the Harvard School of Public Health, they have developed a material, they call it, I forgot the name, some name, it is basically a nano water. So, what they do is they take a water droplet and then they split it up into, like my first question in the test, they split it up into number of particles. One drop of water, split it up, and as the water is split up, it put it under a strong electric field. So, what happens? The size is critical. So, what happens is the oxygen, the H2O, the oxygen of the water, water becomes a very strong oxidizing agent. They have to do it very carefully, because if the particle size increases, it produces too much oxygen, and that oxygen leads to ozone formation. And anybody who is in the area of toxicology would know that we do not want ozone. Ozone is something that is not good for us. I do not know exactly what it does, but it is something that we should avoid. Ozone. So, the particles have to be of a critical size. If it is not nano, if it is bigger size particles, if you put water, in fact, if this is one of the methods of preparing ozone. So, that nano water particles are now sprayed on fruit vegetable surfaces. So, that has a high concentration of oxygen. So, what happens is it oxidizes all the bacteria. There have been tests which shows that the oxygen lyses through the bacteria cells like anything. So, the bacteria is dead. But this is so small, they only need picoliters, because you know there are small drops of water. So, you do not even know that you are spraying water on the materials. They are right now working on that to kind of create, when people travel in a train, people travel in a plane, the person next to you sneezes, next to you have an infection, and it carries over. You do not want to catch an infection of another person. So, they are on the process of commercializing a spray. So, whenever you go, like perfume, you would be spraying this material. So, you will create an artificially charged oxygen surface around you, and no bacteria would be able to come in through there, because they would be always lysed there. And they are also working with the same agent, they will put it in the air conditioning system. So, when the air conditioning air comes in, it would come out with that. So, these are the good parts, and since it is water, there are no metallic nanoparticles are the worst culprits. So, if you can stay away from metallic nanoparticles, you are okay. So, it's water. Only thing is you have to be concerned is you are not making ozone. If you are making ozone, then that would be. It's talking about different types of is working on this this grant which we worked on, the addressing the critical and emerging food safety issues. So, what was the experiment conducted? There were 12 plant species which were taken: corn, soybean, wheat, alfalfa. Again, this was chosen by Dr. White. He chose all these. The reason all these were chosen, I think he said some seeds are monocots and some seeds are dicots of this I do not know which one are monocot and which one are dicot, but people in plant would be able to know. Different nanoparticles were tried out. We were tried with single and multi-wall carbon nanotubes, fullerene, silver, copper oxide, silicon, zinc oxide, gold, titanium, and a bunch of other oxides. Again, these particles were, why these were chosen, these were things that are more available, which are on the nature. Is the reason is copper. If you have copper nanoparticles, copper tends to change down into copper oxide. So, it is more likely that you will be having these oxide nanoparticles. So, we did experiments on hydroponics. Why did the experiment? Hydroponic means, for people who are not in the area of doing experiments on with plants, it is you give the plant survives on just water. So, you do not apply any soil to it. The reason why we did not do a soil puts a great degree of uncertainty. Soil itself has silica nanoparticles. It has other elements over there, and it gets very difficult to monitor what is happening. Currently, people are doing a lot of experiments with soil, but you have to be careful in trying to see what exactly you are trying to monitor, because in the number of unknowns become increased. So, that is why it was done hydroponic. So, how was the toxicity? Mentioned. So, we were trying to find the impact of silver nanoparticles, bulk particles or ions on wheat, biomass we are trying to find the biomass. So, how was the experiments done? The plants were put on hydroponics. You see their plants are put inside, are water given with nutrients, and then what was it was these are the days of exposure, and on this side, can't read it very well, why it's written in yellow. This is in one experiment we studied different parts. We studied roots, we studied shoots. So, how much the roots grew, so it was monitored, and so this is how the roots grew, and sorry, the red is how the roots, and this is how the shoots. So, the nanoparticles were administered, and they were taking in. There was one experiment where we did it without the nanoparticles. The other is then one of the reasons we had to also look for nanoparticles is it was given along with the to survive. There are certain nutrient elements which have to be added to the plants, and again we have to see whether those have a role with the nanoparticle. This is the experiment I was mentioning some time back. Is we tried to make the nanoparticles in suspension, because if you put the nanoparticles in the water and then at the end the nanoparticles all settle down at the bottom, then you come up with conclusion that that it's not toxic. It's not a correct experiment, because the nanoparticles haven't gone into the plants, and you can't keep it sonicating all the time. That would actually have a harmful effect on the plants, and the plants anyway would then not grow how much they would. So, that is the time we tried to use SDS, and we found out that everything was coming out toxic. Later we found out the toxicity was caused by SDS and not by the nanoparticles. So, this is we had the impact on the shoot. So, again looking at this data, what do we observe? Again I can't read the scale. So, we were measuring the silver content. This is the silver content we are measuring, and as we look at the number of, I think, days over here, we see the silver content change. So, days of exposure, and the cumulative, the other things what we measured the transpiration, we measured the weight. So, the weight of the plants, but if you keep weighing the plants, you have to make one of the allowance that the plants have to transpire. There would be water that would come out from the leaves and they would go. So, there has to be a kind of a equilibrium. So, based on that, if you take that equilibrium off, that is, there are different ways of doing that. Once you take that thing off, then only you can say that the plant is growing in weight. Otherwise, if you keep the plant and then weigh it, whatever water you have supplied, you put it over there, a part of the water kind of gets transpired away, and so that is, we made allowance for that. We did. So, days of exposure, let us come over to, we observed that copper oxide nanoparticles, this, you look at the data over here, this is our control sample, the white, and the growth on this side. Once we were doing at biomass, we looked at roots, we studied the roots, we studied the biomass, and we studied also the shoot length, the stem length, and again these plants have a growth time of about, I think, like 20 days, like 14 days or 20 days, and we kept monitoring it every then. And we observed this white line. You compare it with the white line with all these are with different concentrations. We tried with different concentrations, and if this is the control, so this does not have any nanoparticles, and compared to the control, you look at these graphs, and you would be getting an idea. So, anything that is below the graph, it is toxic, but then again there has to be statistically significant. If it falls under the statistically significant area, then you cannot definitely say it is toxic. So, these was silver, I think this is silver. When we did it with copper oxide was turning out is extremely toxic. So, that is we, in fact, with copper oxide, the data what we have is not present over. We did not put the whole data. As we change the concentration, what would you expect? If you increase the copper oxide more and more and more, your toxicity should increase. But what we observed was, here we get it to a certain extreme, but if we increase the concentration, we observe that actually after a certain point, if you add more concentration the toxicity in fact decreases. Copper oxide has a critical concentration, critical density at which it is the most toxic. In fact, dilute copper is more toxic than dilute nanoparticle of copper oxide is more toxic than the more concentrated ones. In fact, there are other studies which also we did an experiment on nanofluid. Nanofluid means you are trying to measure the conductance. You apply copper particles in the fluid to make the conductance increase. There is a critical thing. So, it is basically, any one of you familiar with homeopathic medicine? Homeopathic medicine means dilute concentration of something works better to you. In fact, from nanotechnology we observe that in fact dilute concentration has a better effect than more concentrated concentration for certain, at least for copper oxide that is the case. So, dilute copper oxide is more toxic than concentrated copper oxide. The reason is copper forms an ion. If you look at the chemistry, you would know copper forms a particular ion. So, when you are putting more concentrated of it, it screens one from the other. So, that is why the toxicity is kind of screened out. If you have dilute concentrations of copper oxide, that ion has not formed that much. So, the screening effect is less. So, there are two effects which are competing against each other, the screening effect and the nanoparticle effect. We did the experiment with multi-wall carbon nanotube and activated carbon. This is activated carbon. So, you see the multi-wall carbon nanotube is slightly more toxic, and this is the, again these effects are, we are trying with different one. This is with zucchini. This is one of the issues which we had is we tried different bunch of plants and there was no consistent saying that multi-wall nanotube is toxic in certain plants, multi-wall nanotube was not as toxic with the other ones. So, this data was presented. Dr. White is from the toxicology, his area of specialization is toxicology. So, he presents data which are more toxic because that is what gives him, that is what he is interested on showing. So, the data what is presented are the ones which are the ones which show more toxicity. So, this is fullerenes. When we apply fullerenes compared to activated carbon, you observe is that actually fullerenes are performing slightly better than the control. So, fullerenes is acting like a fertilizer. Multi-wall nanotube is more toxic. There is a speculation why that happens. There have been other results also where people have done experiments with tomato and other plants, and they have found out what is, first of all, fullerenes do not have the metallic nanoparticles that the carbon nanotube, multi-wall carbon nanotube has. So, the effect what you are observing over here is not only just multi-wall carbon nanotube, but also you have those metallic catalyst particles. You cannot totally avoid them unless you choose a method by which multi-wall carbon nanotube has been produced without the catalyst. But the downside of that is if you choose, we chose the material of multi-wall carbon nanotube or even single wall carbon nanotube which is not made with the catalyst, but the main problem of that was it is not soluble in water. So, we could not do any tests, the thing was all settling down at the bottom. So, it did not have metal particles, but it was not going into the plants. So, we could not there were we thought about injecting it into the plant surface to into the plant to see the effect of it, but we intentionally did not do it because what we were trying to show is the effect of the nanoparticles in the environment. If we inject it, we would definitely find it more toxic, but that information is not pertinent to us because the plants won't be here, as I am saying, is my friend told is for the vegetables they are injecting the nanoparticles, that is a special case, but normally the nanoparticles won't be injected into the plant, they will be coming in from the nature, from other. So, that is number one, and the second thing is it is the nutrients and all, it is a hollow space. So, it can be used as a drug delivery type mechanism. So, where the nutrients can maybe soaked in inside and then the nanoparticles get in over there. So, the plants have a better way of how the nutrients kind of reach there. So, that is a speculation. We have been able to try to observe that by taking images, AFM scans of the shoot as it goes up. We have not got any conclusive results of getting that. This experiments was effect of nanoparticles, copper oxide on corn germination and root elongation. So, as again the other thing what we observed, what we were trying to see is how many of the seeds germinate when we put it in a solution of copper oxide nanoparticles. We also found that toxicity shows over there. The number of seeds germinating is less for the copper. If you look at over there, this is how many of the nanoparticles these and the total number of total amount of copper that is present in the root. So, the root uptake of copper nanoparticles, we see what is happening to the cells. So, these this part I was not involved, but it was a part of the project. Which says of the functionality of upregulated. I miss. Okay, so they were trying to find out the effect of what happens to the genes depending on with the nanoparticles. So, why does the plant grow taller, or why does the plant become toxic? Is obviously because the nanoparticles kind of change some of the genes. Some genes are upregulated, they perform more. Some genes are downregulated, perform less. So, this was an experiment which was performed to see which nanoparticles have effect on which type of genes. Again, I am not an expert on this, but that experiment, these were the different types of genes, and they were trying to compare the effect when they used titanium oxide. These were the genes which was upregulated. Zinc oxide, these were the genes which was upregulated. So, these genes performed more than the other one. So, what was the mechanism of silver nanoparticle toxicity and tolerant? So, so it is they looked at this particular, again I do not know what plant, I think it is spinach. We did the experiment, either lettuce or spinach. Anybody who knows what is this? Gurpreet, you know what this is? Okay, I am asking him because he's a plant biologist, so that's the reason I asked him. Anyway, we did experiments on spinach. I know that. I don't know what this one was. So, upregulated, downregulated. So, with the silver nanoparticles putting in, they were trying to find which genes get the controlled different concentrations of, this is silver nanoparticle, and then it was compared with silver nitrate. What happened to the biomass of the plant when we applied different values of silver? You see, there are these three different again the type of upregulated. So, when these things go up, those this is a normal control piece, and as we apply the nanoparticles, this is the reason why it gets toxic. So, more data, this is the data that happens on the shoots, this is the same thing that happens on the roots. What we are observing is that same genes, when you are what you are looking for, they behave differently on different parts of the plant. So, if you are looking for the shoot and if you are looking for the root, these say for example, when you put the silver nanoparticles in the shoot, this whatever this ECS 16.5, this gets regulated more, while when you are looking it for the roots, it is this one which has gets more. So, the fourth objective, we want to find the interaction of nanoparticles with coexisting organic chemicals. Again, pure nanoparticles are never added to the plants. It's always come as a group, as a fertilizer. So, that is what we would like to do. So, we wanted to find the interaction between nanomaterials with these coexisting contaminants. Again, could the bioavailability of the legacy pesticides be affected? Could the intentional pesticides be affected? So, intentional pesticides which people are putting in, and then there are some of these which are already there. I had written a proposal for the Department of Agriculture. Some countries are very much concerned with this. One of I am at least for me, one of the most greenest country, that was New Zealand. I had written a proposal for the Department of Agriculture, Government of New Zealand, trying to use my sensor to monitor some of the reason the grant was not funded was I did not have a proper way to what happens after once the sensing is done, what happens to the sensor after, whether it lands up finally in the water stream or something. So, you have to have any proposal you write from any branch, you have to have a very good idea of what happens and make sure that it never lands up into the groundwater stream. So, some countries are very much, the other countries do not care about it that much. So, so that this issue was being able to address. So, the objective is to quantify the uptake of the pesticides through the nanomaterial chemical interactions. The chemicals which we add more are this humic acid. Humic acid is basic because we are doing experiments on just hydroponic, just water. We need to give it some properties of the soil, and humic acid is something which is anybody working, it's found from the component of the soil. It's the main component of the soil. So, we wanted to see how the nanoparticles interact with humic acid. That was one of the concerns. Is again, it's not actually doing something to the plants. It is somehow it is working something with the humic acid. Even when you go hydroponic, it's not just you're adding the nanoparticles to the water and doing the experiment. The water also has this humic acid and other components, and so that's. So, with zucchini, with the shoots, we did the experiments with the nanoparticles with fullerene, the fullerenes plus humic acid, and just this DDE is also another solution like humic acid. I don't remember the full name of it. Anybody knows what is DDE? It's something which is added to the hydroponics. So, anyway, so we had DDE, we had and humic acid, and we were studying the effect of the fullerenes. So, when you see just DDE, it changes. This is the time when we didn't present its data, but it's negative. But when we looked at with SDS, that is when we found out that is. So, the toxicity could be caused by the nanoparticles going in and doing some change to the chemical, one of these chemicals, and that is kind of having an effect, or the toxicity could be caused by for silver nanoparticles, it's being caused by upregulating and downregulating the genes. So, again, once we know the cause of toxicity, we can address it. So, that was the whole idea. So, this is we did it with the roots. And so, what was the conclusion? This is the main material. When you add it with the fullerene, you see the material itself, the total when we add, you see what happens for the shoots. When we have the DDE plus humic acid, then the amount of DDE when we add it with the nanoparticles, it is more than the pure one, but so the fullerenes. So these are the conclusion, the zucchini shoot, root, and the total. So the fullerenes enhance the DDE accumulation in both roots and shoots. So the fullerenes, again, the reason we were trying to study this was to see why the good effect of the fullerenes is. So suggests that the interaction between the DDE and the fullerene, because you see when you put in with the fullerene, this is the DDE percentage, it goes up, and the fullerenes clearly present in the roots and on the same experiment done with tomatoes. So with the tomatoes, fullerenes enhance DD accumulation in most likely on roots, these values. So I think DD is
It's a pesticide just like a DDT. Dichloro, diphenyl, trichloroethane is DDT.
yes.